Hello everyone, welcome to my channel Capsa. Today we will learn about antibody or the structure of antibody. Okay. So what is antibody? Okay. Actually, antibody is a Y-shaped protein made by plasma cells in response to an antigen. Okay. So it is a Y-shaped protein that is made by plasma cells in response to antigen. It is also known as immunoglobulins. Now, what is antigen? Short form is IG, okay, immunoglobulin. Now, what is antigen? Antigen actually are the foreign substances that causes the body to make an immune response. So, antigen are the foreign substance. So, when there is an antigen present in the bloodstream, antibody detect it and originate an immune response. Okay. Now we need to be clear about who synthesize the immunoglobulins or antibodies and from where antibodies are secreted. So actually antibody synthesized by B cells, okay, or B lymphocyte cells, and it is secreted by an ACS cell or also known as antigen-secreting cells. Now, what is antibody-secreting cells? Now, what is antibody-secreting cells? Antibody-secreting cells are differentiated form of B cells or B cells converted or differentiated to form antibody-secreting cells. Okay, clear? Now, we will need to clear about the structure. Okay. So, what is the structure of immunoglobulin or antibody? This is the structure of antibody. I have drawn previously for time-consuming purpose. You can see the violet part are antigens. Okay, and these two are light chains. Okay, and the total is first one and the second one are heavy chain. Okay. And this region is known as Hinge region. You can see here. These are the disulfide bond. Two sulfur molecule form each other. And this part, brown part is known as variable region or FV, from variable fragment sometimes. And the orange part is known as constant region or FC, constant fragment. Okay. Now, you can see here that the yellow portion or the two light chain is known as FAB. FAB, it means antigen-binding fragments. Okay. Now we need to be clear about what is the structure. Okay. So, antibody actually consists of 4 polypeptide chain and variable region is composed of amino acid that is 110 to 130. 4 polypeptide chain. Okay, 4 polypeptide chain means 2 light chain, 2 heavy chain. Okay, you can see here 2 light chain I have denoted and 2 heavy chain. Okay, now these polypeptide chain are the main composition of antibody. In human, actually, light chain can be one of these 2 classes. One is kappa and another one is lambda. And both of these light chain have 2 domain. One is constant domain or constant region and another one is variable domain or variable region. You should keep in mind that one light chain plus one heavy chain and another one light chain and another one heavy chain forms heterodimer. So what I have said, one light chain, take one light chain and one heavy chain, total one heavy chain, another one light chain and one heavy chain. These 2 forms heterodimer. Clear to you? Now, 2 heavy chain forms heterotetramer. Dimer was 2 molecule and tetramer means 4 molecule involved, or here 4 polypeptide chain involved. So, tetramer means 1, 2, 3 and 4. These 4 are forms heterotetramer. Clear? Now we need to be clear about what is the function of these fragments. That is important. Okay? So, FC or constant fragment, a constant region, actually responsible for its biological activity. Okay, now we need to be clear about how they shows the biological activity. Actually, they bind with the FCR receptor, okay, or also known as constant fragment receptor. Thus, after binding, this shows biological activity or bioactivity. Now, what is the function of FAB or Antigen-Binding Fragment? Actually, it determines the antigen specificity and enable it to bind with the fragment. It means antigen, to enable the antigen to bind with it. Okay, so it is responsible for antigen specificity. So this is the end of my lecture. If you like my video, please do like, comment, share and subscribe. Thank you for your attention.